Thanks for the organizers' invitation. Here is Pao Yuan from Beijing, China. And before my presentation, I need to close my camera to maintain the nerve stability. Okay, my presentation may be a little different from the previous speakers. My talk mainly focus on the basic research. So my topic today is microglia glucose metabolism disorder in Alzheimer's disease. Okay, previous speakers had given many backgrounds about AD, and we know this disease is characterized by two classic pathological hallmarks, beta amyloid plaques and the neurofibrillary tangles outside the neurons because of the hyperphosphorylation of tau protein. However, the pathogenesis of AD is very complicated, and there are many hypotheses of AD, including a beta cascades, tau hyperphosphorylation, oxidative stress, mitochondrial dysfunction, and metabolism disorder, neuroinflammation. Of course, our lab mainly focus on these 2 points in recent years. Okay, microglia are the resident immune cells in the central nervous system and plays very important roles during the brain development and the disease progress. They are involved in many biological processes in the brain, such as neurogenesis, synapse planning, myelination, phagocytosis, inflammation, and so on. So the normal function of microglia are very important for maintaining the brain homeostasis. In fact, there are many reports that have shown that microglia are involved in the pathogenesis of AD. This slide shows you here that many AD risk genes are associated with with microglia, including the very famous gene TRIM2, as well as CR1, CR33, because the mutation of these genes will increase the prevalence of AD. But we also need to be aware of the heterogeneity of microglia. Several microglia subpopulations have been reported, and each subset of different functions. For example, M1 microglia mediate the neuroinflammation and promotes AD progress. However, the M2 microglia can phagocytose the A-beta and exert the neuroprotective effects. So to manipulate the microglia phenotype transition is potential therapeutic approach for AD treatment. From the macrophage, we think that the metabolic reprogramming may determine the phenotype transition in the microglia. We can see on this slide that during the prolonged inflammation activation of macrophage, these cells will switch metabolism from the mitochondrial oxidative phosphorylation to the glucolysis. However, when they are under M2 activation, macrophage mainly use the mitochondrial oxidative phosphorylation for the energy production. Similarly, a recent paper in the in the Science Translational Medicine has demonstrated that microglia activation or neuroinflammation is very positive associated with the microglia glycolysis. as indicated by the glucose uptake here. We can see in these slides, we can know that in the clinical evidence also show that the end product of glycolysis lactate is also significantly increased in the CSF, the sample of the AD patient when compared to the control samples. This evidence indicates that the activation of the glycolytic activity in microglia may contribute to the elevated lactate levels in the AD brains. Actually, lactate is not a waste product anymore. It has many functional aspects in health and diseases. For example, it can serve as an energy source for neurons. Second, it can also regulate tumor immune microenvironment and to exert immunosuppression. Furthermore, it can also bind to the target proteins such as MARVs to modulate the signaling transduction. More recently, lactate was demonstrated to serve as a substrate for histone lactylation, thus participate in the regulation of the gene transcription. So our scientific question is that whether lactate metabolism is involved in AD pathogenesis and how lactate affect microglia functions during the AD development. To answer these questions, we first examine the lactate levels in the brain tissues of the wild-type and AD models. As hepatic, we can see here, we found that the lactate levels are significantly increased in the AD mice compared to the wild-type control. Next, we purified the histone proteins for histone lactylation analysis, and we found that the novel epigenetic modification is also increased in the AD mice brain, including the cortex and hippocampus. Particularly, we found one of the mass H4K12 lactylation is the most affected site. Interestingly, we observe similar results in the human AD brain samples, and this the codificate the data also confirm these results. Next, to examine the changes in the H4K12 lactylation in different cell types in the brain, we next perform co-staining of the H4K12 lactylation along with antibodies against markers for neurons, neuron, and astrocytes as GFAP here. And we found that no obvious difference of the H4K12 lactylation intensity in neurons or in the astrocytes between the wild type and the AD model mice. However, we observed that the H4K12 lactylation, the intensity of this H4K12 lactylation increased significantly in the microglia, especially in the microglia associated with the A-beta plaque. We also purify the microglia and detect the point H4K12 and other size also increase significantly in the AD microglia. We know that the histone modification can affect the transcription of the target genes. So to explore the potential functional and significance of the H4K12 lactylation and microglia in AD, we perform genome-wide CAR-Tag analysis here to identify the candidate genes that are regulated by the H4K12 lactylation in microglia. The results show that very obvious enrichments of H4K12Ps in the myoglia of AD brains, with most of the binding Ps located within the promoter sequence. And the KEGG pathway analysis so that these differential genes are mainly enriched in the HIF-alpha pathway, toll-like receptor, NF-κB, TNF-alpha signaling pathways that are involved in glycolysis and inflammation. Specifically, the core Ps identify candidate genomical loci at glycolytic genes, including the HIF-alpha, PKM, and LDHA. Similarly, we performed the Q-chip analysis, also indicate that the H4K12 latylation levels on the HIF-alpha and PKM, LDH promoters were significantly elevated in myoglia isolated from the AD mice compared to the wild-type mice. Consistently, the qPCR analysis so that the expression levels of these genes are significantly elevated in the AD myoglia. We next examine the histone latylations in the time course data sets of the isolated myoglia from the different ages of AD and wild-type mice. Interestingly, we noted that the levels of H4K12 latylation levels increases in an age-dependent manner in the AD myoglia. Importantly, we observed that concomitant significant increases of these genes, messenger RNA, as the AD pathogenesis progress in AD myoglia. Therefore, based on these findings, we propose there may be a positive feedback loop between the glycolysis histone latylation that propagates the metabolic switch that I mentioned before in the myoglia of the AD brain. So next, to experimental confirm the functional impact of the glycolysis histone latylation PKM2 positive feedback loop on myoglia. We constructed a PKM2 stable knockdown cell line in the cultural myoglia BV2 cells. We found that knockdown of PKM2 resulted in a dramatic decrease in latte production and both pan histone latylation and site-specific H4K12 latylation. Importantly, the Q-chip analysis so that the H4K12 latylation levels on the promoters of these glycolytic genes, including the HIF-alpha, PKM, and LDHA, was significantly decreased in the PKM2 knockdown cell lines compared to the control cells. Consistently, with our early observations of that H4K12 latylation mediate the transcriptional activation, the qPCR analysis so that the knockdown of PKM2 leads to the significant decrease of the messenger RNA of HIF-alpha and LDHA. Additionally, we also use PKM2 specific inhibitors to perform similar experiments and the results so that these inhibitors also mimic the phenotypes of the PKM2 knockdown experiments. Next, to investigate effects of these glycolysis histone latylation PKM2 positive feedback loop on AD pathology. We close the PKM2 floss mice with the myoglia specific query, CX3, CR1 query here, and in the background of the AD, and we gave the mice tamoxifen to induce this gene deletion. And we can see here, after knockout of PKM2, the latte production significant decrease in the myoglia isolated from the mice, and also the histone lactylations also decrease, especially in myoglia around the A-beta plaque. Next, we co-state myoglia with antibodies against PKM2 and against A-beta to examine the effect of PKM2 on microglia activation. We can see here microglia with strong PKM2 signals in AD brain sections show abnormal activation as indicated by the enlarged cell bodies and shortened process. However, these phenotypes can be rescued by knocking out the PKM2 in myoglia. Here, so you here in F, the Western blot analysis also confirmed that after knocking out the PKM2 leads to reduce the microgliasis as indicated by the reduce of the IBA1 protein. In addition, we also found that the homeostasis of the myoglia was rescued by knocking out PKM2 as indicated by the specific marker, homeostatic marker TMEM119. Here we can see here in the AD brain myoglia almost lost the TMEM119 signals. However, in the CKO AD mice, myoglia partially express these homeostatic genes. Additionally, we also performed the myoglia isolation and detect related message RNA of the homeostasis genes and pro-inflammation genes. We can see here they confirm the results from the co-staining data. Next, we also use TS stain to examine the A-beta plants in the brain sections of AD brains and the CKO AD brains to investigate whether blockage of this positive feedback loop may affect the A-beta pathology. The results show here, we can see that after knocking out the PKM2, the A-beta burden was significantly reduced in the CKO AD mice brain. Okay, here, so here, this is the quantified data. Additionally, our Western blotting analysis also confirmed that the A-beta levels was significantly reduced in the CKO AD mice. Importantly, we perform Morris water maze to detect the spatial learning and memory abilities and found that knockout PKM2 to blockage of the PKM2, the glycolysis, histone methylation, PKM2 positive feedback loop, and found that when knockout PKM2 can significantly improve the spatial learning and memory of the AD mice. So Finally, we see to test whether pharmacology inhibition of PKM2 may confer any protection against AD. We gave the AD mice with the PKM2 inhibitors, as shown here, sequonin and compound 3Ks every other day for 1 month. And after that, the brains of the mice were analyzed for AD-related pathologies. And the results show that PKM2 inhibition attenuates AD-related pathologies, including the myoglia activation and the A-beta burden. So here is our model. We propose that based on our findings, we found that in AD myoglia, they are display an activation of the glucolytic phenotype, and after activation of this glucolysis, it will lead to the significant increase of the lactate production and the histone latylation. After that, we further found that this histone latylation could promote the transcription of the glucolytic genes, such as the PKM2, LDHA, so thus forming a glucolysis, lactate, histone latylation, glucolysis positive feedback loop in myoglia. And we found that this loop will exacerbate the dysfunction of myoglia and AD pathologies, and finally the cognitive decline in AD model mice. Through genetic or pharmacology inhibition of PKM2 to block the glucolysis, latylation glucolysis loop can rescue the homeostasis of myoglia and reduce neuroinflammation, as well as other AD-related pathologies and cognitive decline. So after publication, our work also receives attention by other journals and were highlighted by Nature Chemical Biology and molecular neurodegeneration. Our work. Okay, finally, I want to thank Professor Zheng Qiangyuan and the Professor Lu Yangshun for directing the project. I also want to thank my cooperator, Dr. Lin He from PKU, and Dr. Zhang Jing, and many fundings here. Okay, thank you.